Hello students, welcome to the theory class of Principles of Plant Breeding. Today's topic is on breeding methods of self-pollinated crop plants, which includes pedigree method, bulk breeding method, and single seed descent method. Before we start the lecture, it's important to understand some of the important concepts here. The difference between natural selection and artificial selection. So in natural selection, we are going to select based on the fitness of the individuals. From a heterogeneous population such as this, we are going to select individuals which can survive through harsh conditions of biotic and abiotic stresses. Well, in case of artificial selection, we are going to select based on the economic yield or based on the welfare of the human being. So from the evolution point of view, so human beings started selecting those genotypes which are useful to them, and so these are the two difference between natural selection and artificial selection. So in the latter plant breeding programs, we are going to imply natural selection and artificial selection, and it's important to clear the concept of natural selection and artificial selection. And another concept is the variation which is required in the plant breeding program. So for every plant breeding program, variation is a must. Either it should be existed well before the plant breeding program starts, and either if it is not there in a population as such, uniform population as such, we have to create. So we have here two populations, one being uniform and another which is heterogeneous or segregating, you can call. So there is little room for improvement in this uniform population, while in the population as such, you can select those individuals which are superior and those reject those individuals which are inferior. So for plant breeding program, we need such populations and we don't need such populations. We need this as a resultant, but we don't need this as a base population of plant breeding program. So if there is no variation as such, we have to create variation by either hybridization, mutation, polyploidy, semiclonal engineering. In today's lecture, we're going to study the plant breeding methods which are created by variation and later followed by selection. So by the end of this lecture, you'll be able to answer what are the breeding methods of handling segregating generations. You will be able to understand the pedigree method, the bulk breeding method, and single seed descent method, and you'll be able to answer the advantages and disadvantages of these methods. And what is handling segregating generations? And segregating generations is supposed to mean the F2 and further generations like F3 and F4 generations, because in these generations these individuals are the genetic components of parent 1 and 2 are segregated in the whole populations according to the second Mendel of inheritance. And these these populations, these segregating populations are usually uh, the favorite populations from which we as a plant breeder we are going to select individuals. And breeding method which can be done, selection can be done in these segregating generations are pedigree method, bulk method, and single seed descent method. So the first method is pedigree method, which was outlined by Love in 1927. In this, individual plants are selected from F2 and subsequent generations, and the progenies are grown in the next generation, and it involves record keeping of parent and offspring relationship, and it continues till the progeny becomes homozygous and they shows no segregation at all, which is at later stage F7 to F8 generations. And in case of pedigree record, we have to maintain record as such, which includes data of the individual's plant height also, and number of accession number of the individual of the selected plants, of each and every individual plants of each and every uh, segregating generations. So coming to the process of the pedigree method of breeding, the first and foremost important thing is the selection of parents. What type of parents we need to select? The type of parents that we need to select are both agronomically superior parents, which are diverse and which can give as much amount of variation in F2 and segregating generations. To be remembered, the type of selection, the type of parents that we usually select in backcross breeding program is totally different. Those are recurrent parent and donor parent. Recurrent parent being agronomically superior, while in case of donor parent, which is having uh, inferior agronomic character but having the desirable genes. In backcross breeding program, the intent is to transfer the desirable genes into the agronomically superior ones. While in this case, in case of pedigree method, the intent, the goal and objective is to transfer both these desirable characters into all the F2 populations and selection of plants which have all the differential components of the P1 and P2 genotype. So let's start the plant breeding program. First, there will be crossing between parents, and then there will be development of F1 populations. F1 10 to 30 seeds of F1 will be space planted, and seeds will be harvested in bulk. And all these seeds which are developed from all plants are going to be grown in the next generation, which is F2 generations. And F2 generations, to be remembered, is grown in a large population, as large as possible, and it should be in a ratio, in a proportion that the F3 plants to be selected is 5% of the F2 populations. And how much amount of F3 population is required depends on the type of parents that we use in pedigree method. What kind of parents? If the parents are closely related, the amount of F3 population is less, while if the parents P1 and P2 are diverse, the amount of F3 population is huge. So if we select 100 seeds, it basically means there is a comparatively close relationship between P1 and P2 compared to 500 plants which we are going to select, maybe because of the parents are diverse. So if we select 100 plants from 2000 plants and 500 plants from 10,000 plants, the proportion is 5%. 5% of plants are going to be selected from the F2 population. And what kind of selection is to be done? Progeny selection is to be done. And what is progeny selection? We have already learned in the previous class, which was discovered by Johansson. So each and every superior line is selected from these F2 generations, and it is grown as individual plant progeny in F3. That means all the seeds from a single selected individual is grown into a row of plants. Okay, and from this single row of plants we are going to select plants, okay, individual plants which are superior in this particular row of plant. Okay, superior plant selected from superior progenies. From this single line of single row of progenies, we are going to select superior ones and proceed to next generation and so on. Okay, so it has to be proceeded up to F4 generations, and later on in F4 to F5 generations, single plants has to be selected, superior plants has to be selected from superior progenies and grow multi-row plots. Okay, multi-row plots here, three row plots are given, which are grown from a single uh, a single row as such. Okay, single plant as such, which are given here. If we select the first individual from the first row, and we grow these multiple rows, so we have a multiple row block as such, and second and third from the fourth or fifth individuals which are going to be selected from each rows. And superior again from F5 generations, the process of selection should be in such manner that if the individuals shows no segregations, if all the individuals doesn't show any segregation in this particular plot, is going to be selected and bulked into the next generation as a plot. And if there is a segregation, if there is a variation in this particular plot, so those individuals which shows homozygosity or those with homogeneity will have to be selected only, and those which are superior will have to be selected only, and we'll have to reject the inferior ones into F6 generations. And in F6 generations we are going to select those individuals which are showing no segregation at all. Okay, and those individuals which are those plots which are showing little to more amount of segregation, we are going to reject it directly. And those who surpasses this selection process are going to undergo preliminary yield trial. Okay, so in this uh, in this F5 and F6 generations, in every 3 to 4 multi-row plots, okay, we are going to have a parental line rows, okay, in order to have a comparison between the these lines and the parent line, okay, and select those superior ones. And F6 also the same thing. And in F7 generations, the selected lines are going to be tested against a control in replicates, in several replicates, 4 replicates here, and quality test is started here. Yield and yield related traits, and at the same time disease insect resistance are can be proceeded from here. And in the next generations, if it if it succeeded in preliminary yield trial, it can go through multi-locational trial, or also known as coordinated yield trial, or also known as advanced yield trials. So this after passing through a preliminary yield trial, so it has to be shown in different locations of AICRP research stations, or either in farmers field, in order to test against the test for the adaptability against the checked varieties of the particular regions. Okay, 4 different regions are indicated here with 3 replicates. So 2 to 5 superior lines which are selected from these are going to multiply and then these multiplied seeds are called as breeder seeds, which are going to be which are going to be developed into foundation seed and then to certified seed for distribution to the farmers. So this whole process of pedigree method can be classified into 3 different phases. The first one is creation of variation, which is a crossing of 2 different parents, diverse parents, and forming F1 and F2 generations. Second one is developing, testing, and selecting potential cultivar, which includes progeny selection and selection on quantitative characters, the yield characters, and, and finally testing for experimental varieties. Say for example, for finally when the varieties are confronted for homozygosity, they have to be tested for adaptability and preliminary yield trials and coordinated yield trials and then release. So some modifications can be done for the convenience of the plant breeder and the situations. So the first modification can be in F2 generations, only few plants can be selected, which reduces the effort to produce all these lines. Okay, superior plants which are found in F2 and F3 generations can be selected, and which reduces much of the effort which involves in selfing or inbreeding of all these generations. Okay, which is known as early generation selection. The second modification which can be done is the progeny selection can be done in only one step. Progeny selection is done only from F2 generation to F3 generations, and the rest of the generations are bulked to produce uh, um, huge bulking generation, mass selection. So in this, there is no selection pressure at all, right, after F3 population. And next one is mass pedigree method, which is also known as bulk method. So in this method, uh, selection is not imposed in the early early generations like F2, F3, F4, F5, or F3, even up to F3, F6. Selection is done only in the later generations. So some of the advantages of pedigree method is it is well suited for easily identifiable and highly heritable characters. It takes less time than bulk breeding method, and it is uh, good for easily visible defect progenies with visible defects and weaknesses can be eliminated at the early stage of breeding program, which saves times and resources. So it comes with early generation selection. So it comes with the modification of the first type of modification, early generation selection. Okay, so with this defects such as weaknesses, eliminations, and germination, low vigor and viability can be rejected in the first phase itself. Second thing is the demerits. Maintenance of a pedigree record is time consuming and tedious, and at the same time it is laborious process. It is sometimes impossible when we are maintaining a large F2 population or segregating populations as 10,000, say for example, and it requires skill of the breeder. If the breeder fails to identify the superior individuals and, and undesirable individuals, so the whole process of pedigree method is a failure. And there is no opportunity for natural selection. We haven't bulked it. We haven't imply any natural selection here. All the selections are based on artificial selections. Okay, either it is selection for yield or either it is selection for quality traits such as disease and insect resistance. Okay, next method is bulk method. So it was first proposed by Nielsen LA in 1908 at Swallow, which is a place in Sweden. It is also known as mass pedigree method or population method of breeding. It refers to growing of segregating materials by bulk method when the conditions are unfavorable for selection, and progeny testing when the conditions are favorable. So these are two important process of bulk method of breeding. We are going to hold the selection process when the conditions are unfavorable, and we are going to do progeny testing when the conditions are favorable for selection. So what is this? So in early generations, in F2 and F3 generations, selection for quantitative traits are not favorable because quantitative traits are controlled by polygenic genes. Okay, and selection of polygenic genes are usually advisable, recommended at the later stage from the immortal lines and not from the mortal lines. F2 and F3 generations are called as mortal lines because there is high chances of segregation in the next generation because they are all heterozygous and heterogeneous. So there is huge amount of risk, huge amounts of chances of segregation in the next generation. So selection based on quantitative traits such as yield may not be fruitful. So in case of bulk breeding method, what we have here is bulking until the unfavorable conditions arrives, and when the favorable condition arrives where there is homozygosity, we will select the polygenic traits for plant breeding program. Okay, and first foremost thing is again selection of parents, diverse parents, and forming F1 progeny. Okay, the amount of F1 will be same 10 to 30, and in F2 generation it will be same 2000 plants. And from 2000 plants we are not going to imply any we're not going to apply any selection pressure. We are just going to we're just going to grow the whole population into F3 generations, and grow the whole population of F3 population into F4 generations. Why we are doing so? We are avoiding any loss of genetic variations. Okay, because if we apply any F2, any artificial selection in F2 generation, F3 selection, so there may be chances of loss of those genotypes which may not show its significant desirable characters in F2, but it may show in later generations. Okay, and some of these may be recessive gene characters which are in heterozygous conditions. Okay, and so bulking is done up to F5 generations. Okay, in advanced generations, progeny selections, individual plant progenies are planted from the selected lines of F6 generations. Okay, and then we are going to select superior plants from the progeny rows, and similar to pedigree method we are going to grow multiple row plots from the from the superior plants of the that particular row, and so on. And we have to go for preliminary yield trials and advanced yield trials or coordinated yield trials, and release as a variety. Okay, some of the merits of bulk method are it is simple, convenient, and less expensive when compared to pedigree method. In such, each F2 plant is equally represented till F6 generations. Okay, so there is no loss of variation. Okay, no chances of elimination of good genotypes in early generations, and artificial or natural disease phytotics, winter killing, high temperature, etc. eliminates undesirable types. Okay, so biotic and hepatic stresses also plays a role in natural selection of those genotypes, selection of individuals which can survive, which has fitness for survival. Okay, next thing is long-term bulks are superior than short-term bulks, and it is more advisable and recommendable. And next is uh, since there is no selection process, it is a less attention seeking method. In F2 and later generations there is no selection process, so we have to go each and every generation without selection, and less attention is required. So this is an easier compared to pedigree method, and no pedigree record has to be maintained. This is another advantage which saves time and effort. And in large populations, transgressive segregants are more likely to appear. Okay, what is transgressive segregants? Again, in F2 and F3 generations, those individuals which are superior, which outperforms the parental performance. Okay, parental performance, those are called as F2 transgressive segregants or F3 transgressive segregants. Okay, so, which is different from F1 hybrid variety. They call it heterosis. Here it is called as transgressive segregants. So some of the demerits of bulk method is it takes much time to develop a new variety. Okay, for a natural selection to have an impact on the variety, it should at least take F10 generations. Okay, natural selection of bulking of 10 generation is required, which may sometimes take up to F20 generations of bulking or natural selection, which in case of bulk method of pedigree bulk method, we are only going for 4 to 5 generations of bulking in short term, which is called as short term bulks. So short-term bugs, in short-term bugs, natural selection has little effect. Okay, there is lesser chances of improvement of on the fitness, though there is this is comparatively higher than pedigree method, and there is less scope for breeder as there is no skill to exercise. Okay, any farmer can execute this breeding method. A large number of progeny has to be selected at the end of the bulking period, and a large number of progeny has to be maintained in a huge field plot. Information on the inheritance of character cannot be obtained, which is often available from pedigree method. Since we are not going to maintain a pedigree method, we are not going to have a record of the information of the character that we have selected for. In some cases, at least, natural selection may act as against the agronomically desirable character. Okay. Some during natural selection, we have already discussed that natural selection favors the fitness. Okay, it doesn't go for yield or uh, a character which is preferred for human welfare. Okay, so it may have an impact. The natural selection process, the bulking process, may have an impact on the yield and related characters also. Next modification of pedigree method or bulk breeding method is single seed descent method. In this method, a single seed from each of 1000 or 2000 F2 plant is bulked to raise the F3 generations. Okay, and then this process is continued until F5 to F6 generations, which is F4 and subsequent generations are planted from single seed from each F3 generations, which is again the same process. Okay, until F5 or F6 generations. So each F2 plant is represented in the F5 or F6 generations populations. So what are the coming to the advantage of one of the greatest advantage of bulk breeding method is there is no elimination of any genetic variation in F2. We are not going to have a selection. So single seed descent is a modification of bulk breeding method. So in this case, we are going to maintain the same feature of this bulk breeding method, which is uh, no loss of genetic variance. Okay, so in this process we are going to select each and every seed, one seed from each and every individual, and proceed into the next generation. Okay, we are going to sow only one seed into next generations and not going to sow any more than that. We're not going to multiply. Now, so because of this, the F2 individuals will represent the F6 generations. Okay, but what is the difference if of what is the significance of growing so much amount of generations? So growing so much amount of so much time of so much generation is it increases the homozygosity. Okay, which fixes the gene, and it is much required to be released as a variety. Okay, and during this process inbreeding is done. Okay, each and every individual is inbred, and this heterozygous condition will develop into homozygous condition, the process. So this is a modification of bulk breeding method where all the and natural selection is also executed here. Okay, and there is no loss of variation here. All the individuals are in the F2 are maintained in F5 or F6 generations, and and from F6 generations individual plant progeny is planted, and progeny testing is done from the superior individuals. Okay, or homozygous plants that we usually select from this, and then multi-row plots are grown. From multi-row plots, those plots which show no segregation, we are going to conduct preliminary yield trials in research stations, and, and after preliminary yield trials we are going to go for advanced research trial or coordinated yield trials, which is similar to the process of pedigree method. So for bulk breeding method and single seed descent method, the preliminary yield trial and coordinated yield trial, you can refer from the pedigree method itself because it is well explained here. So some of the advantages of single seed descent method is it is a rapid method of generation. Advanced 2 to 4 generations can be completed in a year. Okay, and it requires little space or little effort and labor, and it makes best use of greenhouse and off-season nursery because to rapidly advance the generations, okay, in 1 year 2 4 generations has to be an advanced, okay, in this method. So it can be possible with the help of greenhouse and off-season nurseries. 2 to 3 nurseries we can grow rabi and kharif. uh, we can grow single type of crop plant in 2 different seasons, kharif, rabi, okay, or maybe in summer also, especially in greenhouse on off-season nursery types. And it ensures that the plant retain the N population like F6 generations, which are similar to F2 populations. Okay, so the genetic constitution, the genetic variation which is there in F2 population is maintained till F6 generation. So some of the advantages of SSD method are it does not permit any form of selection, and and during the segregation generation, and in such a successive generations, the population size become progressively smaller. Why the population size becomes smaller? In while proceeding while selecting single seed from a single plant and moving on to the next generations, okay, there is a risk of loss of this plant, okay, because we don't have a second plant which is representing the first or second individuals, okay, these particular individuals. So if it is accidentally infested by insect, or if it doesn't show germination, and if it fails to uh, some of the genotypes which fails to come up with the optimum amount of germination and viability, vigor and viability, so it won't be able to survive, okay, and because of that the plants or the plot may lost, okay. So there is a risk of loss of plants in this type of in this type of single seed descent method. So this is quite common in pulse plant loss of such type, because in pulses what happens is during the maturity shattering, okay, shattering of pod is one of the most important character of pulses. Shattering means drying of pods and natural opening of the pods after drying itself, and the seed will get fall in the surface itself before we harvest it. So shattering loss is a huge in pulses. So it's coming to the end of the slide. If you have any questions you can ask. Thank you.